Okay, we want to move to the next session. Thank you for coming back, first of all. And then I want to introduce Professor Nava Nava Zisapel from the Tel Aviv University, which actually fits very well this session because she is personally combining the research and development. She was the head of Adams, and she is a really entrepreneur. A woman, I didn't know how we say for women entrepreneur, but a women entrepreneur which combines very nicely. And I want to take this opportunity regarding Tel Aviv University and, following the first very successful sessions that we had, and I want to thank all the speakers, that, another initiative that we are involved in, and actually is very relevant to ALS, is the Human Brain Project, and I just want to mention 3 platforms that could be very relevant to that. First of all, the simulation model and the computerization of all the synapses, the neuromorphing, and the last one is the medical informatics, which Tel Aviv University is part of, we are part of, that really is doing and is very relevant to the next challenge that Prize for Life is going to present. So I wish us all a very successful next session, and Nava, please.
Thank you, Mira, and I'm really glad to be here because I was actually part of the foundation of the ALS, Israel ALS, when the late David started it. And I very much sympathize with the organization, with both of them now, and the challenges that they are actually meeting every day. Now, when we are talking about ALS, now from the clinical perspective or from the therapy perspective, we actually have 2 goals. And this is also the same for Alzheimer or Parkinson or whatever. One is the immediate goal, and that's a symptomatic relief. We want the patients to feel better. The second goal, which is more ambitious, is the long-term goal, which is disease modification. We want to be able to alter the slope of the disease so that even if a patient is being diagnosed at a very early stage when the disabilities are minor, we would like to keep him that way so that he will have a fruitful life and an enjoyable life for like a decade or more, which they cannot afford now to have now. And the problems with achieving that are actually 2 main ones, I think. One is the surrogate markers. We are talking about biomarkers. It's the same in Alzheimer's, same challenges as well. We do need to have some markers for the disease which could be easily measured. We cannot access the brain. We cannot do too much in the brain. Also with Alzheimer, where we do have some now dyes that look at the beta amyloid, we are still far from being able to diagnose the disease with that or even to see a correlation between the change in the amyloid and the memory and the decline in the disease. So we do need these surrogate markers badly because otherwise we are leaning on cognitive parameters which are highly flexible and can, people have looked at it before. I mean, in the last session on Danny Offen's speech, they were asking about the fluctuations that happen with individual patients. And so once having a marker, you can better look at the disease progression or the diagnosis without too much involvement of the emotional involvement of the patient. And then we do need to understand the pattern over time. Whether we have a marker, if we have a marker, it will be better. We can do it much easier and with smaller groups of patients. But even now, when we are talking about a disease and we are coming with a new therapy, we need to know what pattern is expected for the simple reason of trying to guess how many patients we have to include in the trial. in order to see different statistically significant difference between the placebo group and the active arm. And so these initiatives have been already done to some extent with ADNI, with the Alzheimer's disease initiative, and which is an effort which is all over the world. And we hope very much, and it will be then announced later on in the day. Having said that, we will now look into some aspects of these initiatives. First of all, I would like to invite Neta from Price for Life. This is one of the two organizations I mentioned, Price for Life and Israel ALS. They were ensembled in a slightly different way with different goals. So there is a lot of place for both of them, and I am happy that they are both taking part today. So Neta will talk about how can big data drive innovation in ALS. Please.
Thank you so much, Nava, and thank you for Mira Marcus and Uri Asheri for organizing this workshop with us for International ALS Day. In behalf of Prize for Life, I want to also thank all of you for coming and joining us in celebrating ALS research on International ALS Day. And the topic of my talk today would be a switch from the topics that we discussed before. So until now, we talked about drug development and the molecular mechanism underlying ALS. And in this talk, I'm going to talk about a different approach for driving innovation in ALS, and that's through using computational approaches and big data. So I think you all know what ALS is, and Vivian already have given a very good introduction. So most generally, ALS is a neurodegenerative disease that affects the muscles and leads to very fast deterioration in the ability to use the muscles, to paralysis and death. And from our perspective, from Prize for Life perspective, we really want to do everything we can to accelerate the development of treatment for ALS. We were founded in 2006 by Avi Kramer, which I think a lot of people in the room met personally. When he was diagnosed with ALS, he was 29. He's now 40. So he's been living with ALS for 11 years, which make him a very rare sample of an ALS patient. And what he wanted is to really use all of his resources that he gained as a student at Harvard Business School and use them to advance the development of treatment for ALS. And the idea that he had was the idea to use the prize model to attract new minds and new ideas for ALS research. And the prize model really asks what can be done to remove the barriers for developing treatment for ALS. And one thing about this is to really try to be very proactive in finding new areas of development that could be relevant for ALS but are not relevant for now. And one such area of development is quantitative models. So how could we bring all of this data that computer scientists and statisticians have and use that to accelerate treatment of ALS? And that would be the topic of my talk today. So really one of the biggest limitations, if looking at the quantitative perspective, one of the biggest limitations in understanding a rare disease like ALS is the scarcity of data. So ALS affects 1 in 1,000 patients, which might not be so rare, but given its fast progress, at any given moment, there are very few patients alive. So it's estimated there are about 800 patients in Israel right now. And that makes it very hard to do a very large, meaningful study on ALS patients. Now, we saw recently that there has been tremendous development in our ability to aggregate data, to standardize, to store large amounts of data, and they've been extremely relevant to many diseases. And what we really want is to take these advantages and bring them into the field of ALS research, to bring the sort of big data solutions into the field of ALS where they weren't available before. So we have initiatives in different types of data that are listed here, but one I'm going to talk about today is initiatives to bring together clinical data to better understand ALS. So what we wanted is to create a large enough data source for ALS that would enable meaningful research, and then to really encourage a lot of meaningful research on it and meaningful collaborations on it. And I'm going to give examples for how we approach both of these things. So the first thing is creating a large database of ALS data. So we developed a database called the PROACT database, the Pooled Resource Open Access ALS Clinical Trial Database. It's available open access for researchers, so you can get it at www.alsdatabase.org. We developed it in partnership with NEALS, which is an ALS clinical trial consortium, and with the Neurological Clinical Research Institute at MGH. And it was founded by the ALS Therapy Alliance. But what is PROACT? So PROACT is a database of ALS clinical trial, completed ALS clinical trial. And when I say completed, I mean failed clinical trials. So the logic behind this database was that there have been, in the last 25 years, a lot of clinical trials to develop treatments for ALS. And unfortunately, almost all of them have failed. There is only one treatment on the market right now for ALS. So what we wanted is to still, despite the fact that the clinical trials themselves have failed, to have them be used for accelerating research by bringing together and opening them up for clinical research and for quantitative research. So we approached drug companies, we requested the data, we received donations from 23 clinical trials. There are 17 that are already there and 3 that we are adding in these days, and I'm going to talk about them more later. Currently, the database contains over 8,000 records from over 8,000 patients from all over the world, and it contains the type of data that you typically see in clinical trial and in clinical data for ALS. So you have demographic information and clinical information from the clinical visit. So I'm going to talk about a little bit more later. There's family history data about the likelihood of ALS being familial as opposed to sporadic. There's various functional measures such as respiratory rate, blood pressure, and so on. There's lab data from blood tests and urine tests, so a standard blood and urine tests. There's also clinical records from the time of the trial, so any other medication taken, any comorbidities that the patient might have. We aggregated all of this amount of data and we made it open for the ALS research community. And this was approximately 10 times larger than any other database available at the time that we opened it, which was 2.5 years ago in December 2012. So we really wanted to see it spur a lot of research on ALS. And we were very happy to see that indeed it did. So in the last 2.5 years, we received over 500 requests from researchers to use it from 41 countries. A lot of the researchers, of course, come from various academic institutes all over the world. Very important for our goal, we also see people from pharmaceutical companies, even pharmaceutical companies that don't right now have an active program in ALS, which makes us very hopeful to really see a lot more development for ALS programs due to this database. And I'm just going to give you 2 examples of the type of research that you can see being done on the PROACT database, 2 most extreme examples. So the first is a more kind of standard analysis that you would see done on ALS research that finally has been able to be done on a large enough scale to have very conclusive results. So what you see here, these are what is called Kaplan-Meier curves or survival curves. And what you do is you split the population according to whatever feature you're interested in, and you look at the different survival curves for the different splits. For example, you see here that the age of onset of the disease affects survival. So if you had the disease younger, you are more likely to live more with it than if you only had onset of symptom at a later age. One thing that I think is very notable, because the PROACT database enabled to really assess critically a very common hypothesis in ALS research, and that's the effect of weight, or in this case BMI, body mass index. So there is a reverse relationship between BMI and ALS prognosis. Unlike many other diseases that we see in ALS, you can see here that the lower the BMI, so the less weight the patient have, actually indicates poor prognosis. And that's because ALS is associated with wasting and dramatic weight loss. So the ability to really show on a large population that there is an effect of BMI on survival led to the change of recommendation for nutrition for ALS patients to really encourage high-calorie nutrition, and there has been a clinical trial on high-calorie nutrition on ALS due to these results. So it already helped us understand the clinical profile of ALS. The second type of example is the more sort of high-level machine learning approach that we were hoping to see using the database, and that's a research done by Italian research called Roberto Gomeni. And what he did is he tried to take the entire population of ALS patients and see if he could identify distinct subpopulation of patients. What he took as subpopulation is two very distinct clusters, the very slow progressing patient and the very fast progressing patients. And what you could see here is that he was able to meaningfully identify who would be very slow and who would be very fast. And I'll return to that later. So if I'll summarize the most broadly all of the things that we see the PROACT database being used on right now, so we see that it's being used for various scientific exploration, understanding disease natural history and heterogeneity, understanding disease pathways and potential biomarkers through these lab tests and urine tests and comorbidities. And we see that it's also being used by pharmaceutical companies for clinical development, for clinical trials in ALS. So you could use it to simulate a clinical trial, to see how a population in a clinical trial would look like, and to use it to test different hypotheses about this trial. The most broad benefit that we see of the PROACT database is the last point. So what we were really hoping to see is we were hoping to see a lot of people use the database. And through this growth in the workforce of people using it, there would come new ideas and new minds. And we don't want to restrict them to any particular question, but really open it up for their ideas and their creativity about what can be researched on the database. So we wanted to do something proactive to bring all these peoples and all these new minds into the field of ALS research. And as our name is Prize for Life, we typically do it with the prize model. So we developed a prize on the PROACT database. The prize was the ALS Prediction Prize. It was launched in summer 2012, so before the database became available open access. And it invited participants from all over the world to predict ALS progression. More specifically, to use 3 months of clinical trial data in order to predict how the patient is going to look like 9 months later at the end of the year. The prize for this was $25,000. It later increased for $50,000 because we received another donation, which allowed us to give the prize to 2 winners. We did it in collaboration with the DREAM project, which is a project within IBM doing quantitative bioinformatic challenges, and with Incentive, which is a platform for challenges online. And of course, with many, many participants. So before I go into the results of the challenge, I want to go a little more into detail what we asked the participants to do. So what do I mean by measuring progression of ALS? So the common measure for ALS progression is a scale called the ALS Functional Rating Scale. And this scale is a questionnaire given by the clinician to the patient and asking the patient about various daily functions. So you could see here, so one would be speech, so how well can you talk between 0 to 4, how well can you breathe between 0 to 4, how well can you turn in bed, dress yourself, climb stairs, walk, etc. 10 questions, later it was revised to a longer scale with 12 questions. And the possible total scores are between 0 and either 40 or 48. And the general logic behind the scale is that ALS patients all progress differently from each other, so we don't know what would be the next thing that they might happen to them functionally. And Avi said it very clearly Avi Kramer, who started the organization, in a documentary about him. So he had a girlfriend, and she went on an internship for a month. And he was very distraught about that, because as he said, every month I lose something. So by the time she comes back from the internship, I might be able to walk less efficiently, talk less efficiently. I might not be able to use my hands. I don't know what would happen, but I would lose something. So the ALS Functional Rating Scale tries to capture this very broad sense of losing something. So the total number doesn't tell you what happened to the patient, just the general level of his functional capacity. So an example of a patient, as you see in the database, would look something like this. So we have a woman, a patient who is 69 years old. She's Caucasian. She she had her first onset of symptoms 19 months ago. Her weight is stable and normal, which is a good sign. She had bulbar onset, meaning onset in the area of the mouth, which is a bad sign. And if you look at her ALSFRS, you see that as what you expect from bulbar onset, she has difficulties in speaking, and she has some difficulties in swallowing and in salivation. But, for example, the use of her hands is well preserved, the use of her legs is well preserved, and her total score is relatively high for a patient that had already been living with ALS for 19 months. If you look at her lab test and vital signs, so I marked in bold what would appear abnormal. So you see that her cholesterol is a little high. She has some high blood pressure, some triglyceride problem, but everything looks very normal in the lab test. So what we did is we gave cases like this to clinicians. This was one of the cases. We gave 14 cases. And we asked clinician, what would you predict would be the survival of this patient 9 months from now? Where would she be in her disease 9 months from now? And we gave that to 12 clinicians from all over the world, all leaders with their own ALS clinic. And the most common answer that we got is that they would never consider predicting ALS progression with such amount of data. So they would just tell the patient to come back 3 months from now and would not give her any information about her future. But if we force them for this exercise to predict ALS progression, then they vary in results a lot. The most optimistic was actually 0 point lost in 9 months, and the worst one was 11 point lost over 9 months. But what actually happened to this patient is the coming 9 months, she lost no less than 19 points, actually become fully paralyzed in her arms and her legs, and she died a few months later. So this really, I think, captures the difficulty in predicting ALS progression right now. And this difficulty makes it very difficult to treat the patient correctly and plan interventions for them. It makes it very difficult to plan clinical trials, because if you have a patient that looks like a very slow progressing patient and suddenly progress very rapidly, it's very hard to tell if it's something that the treatment caused or it would have happened anyway. So this really highlights the need for better computational tools to understand progression and to predict it better. So this was one of the cases used in the challenge. And we compared so whatever the participants did, they had to outperform what would be a standard method, the baseline method that we developed for the challenge and use support vector machine. And to win the challenge, you had to develop your own method that would be better than this baseline method. And then we opened the challenge for participants from all over the world. We had over 1,000 solvers from 64 countries. So already we felt very successful because we managed to bring to light the needs of ALS clinical research and quantitative research to hundreds of people all over the world. And we surveyed them at the end of the challenge, and we asked them, how much do you know about ALS from 1 to 5? And 80% of them said 1. So they actually were people really outside the field of ALS that we were able to interest, at least in ALS research. Now for the results. So we end up dividing the prize between two teams that scored very similarly. Now, I talked before about how we ask clinicians to predict for the patients. So what you see here, is the comparison between how the methods performed and how the clinicians performed. So this is correlation between what was predicted and reality. So you want this to be as high as possible. This is root mean square of deviation, so the difference between what was predicted and the reality, so you want this to be as low as possible. So the best solutions would be along this axis, very high in correlation, very low in RMSD. And what you saw was that the two winning methods were able to outperform all 12 clinicians. And not only were they able to outperform all 12 clinicians, they actually outperformed all 12 clinicians in all 14 cases. There was not a single case in which the clinician performed better. So this really showed us that these algorithms can have real value in the clinic to be used with ALS patients under the guidance of a clinician. The second thing that was very important to us is that that would also be very relevant for clinical trials, because clinical trials in ALS now work with a lot of heterogeneity and uncertainty about ALS progression. So we worked with a biostatistician from Harvard about how you would simulate a clinical trial during clinical trial planning. And one thing that you need to do when you plan a clinical trial is you need to have a good assessment of the size of the trial. How many patients do you need in order to see an effect? And what we saw that using this model, just by understanding the differences between the patients better, we were able to reduce the number of patients needed by 23%. So a large ALS clinical trial would be about 1,000 patients. So 23% less would mean 230 patients less. So at the most conservative estimate, this would be millions of dollars in reduction in the cost of a clinical trial per every future trial using the algorithms. So we're very happy about that. The algorithms are available open access. The summary of this challenge was published in Nature Biotechnology. The algorithms are available as supplementary information, and the challenge was also covered by Science and Science Translational Medicine. We also were able to identify, very importantly, we also able to identify novel features to predict disease progression. And so some of them were known, and some were features that have never been described before as predicting ALS progression, such as blood pressure and pulse. And for example, from this list, the last one, creatinine, which was very predictive, is now in clinical testing as a potential biomarker for ALS. Another very happy and unexpected development happened after the challenge was closed. So we had two winning teams. One of them were a team from Stanford University, and the other was from a marketing analytics company from Washington, D.C. called Centrana. And they really joined this challenge out of their own personal connection to ALS. And what they decided to do is take the money that they won in the challenge and develop a startup company called Origin Data Sciences. They would use these models, further develop them, and give them as services for ALS clinical trials. And the models that they've developed are now used in 3 ALS clinical trials right now. And they also have adapted them to 12 clinics for free and donated them for these clinics for free. So we're very happy to see now that these algorithms already are being used in ALS clinics and ALS clinical trials. So just to summarize where we stand with the previous challenge, we really saw that if you create a lot of data, then the researchers do come. So if you create meaningful data, you curate it, and you put it available open access, a lot of people come and they do very interesting analysis on it. The other thing that we saw is really the importance of bringing people actively using crowdsourcing. So the ALS Prediction Prize really enabled a lot of people to be exposed to the data and allowed us to guide them to create tools that can really be extremely helpful for ALS research, for ALS clinical development, and for ALS treatment of existing patients. And that brings me to my second topic, which is what to do beyond the ALS Prediction Prize. So one thing that we saw when we looked at the results of the ALS prediction prize, was that we actually saw that the predictions were very similar to each other. So if you look here, you see this is the differences between what the way the ALS actually progressed in the patients and the way the different algorithms predicted ALS progression. So you see that there is some unexplained variability here. However, if you just compare the two winning algorithms, just how do they predict compared to each other, you see a very high correlation. So they actually predicted ALS progression very similarly to each other. And I think that suggests that there's also heterogeneity that isn't captured yet, that isn't understood by any of the algorithms. And the reason for that is that if you ask a machine learning algorithm to predict ALS progression over the entire population, so you have one model for all of the patients, what you see is that the algorithm predicts the best for the most average patients. So the patients that are most common in the database and have the most common disease progression, they would be very well predicted. And as the more unusual patients that are further from the average would be less well predicted. So the very slow progressing and the very fast progressing would not be as well predicted. But of course, to be meaningful for patients, we want all of the different patients to be well predicted, not just the most average patients. And that made us decide to really go and plan another challenge, the ALS Stratification Challenge. And the ALS Stratification Challenge doesn't ask for one model for all the patients. It asks to separate which patients need a different model. So for which patient ALS is the same, and for which ALS patient ALS is different. So we ask participants to identify meaningful subgroups of patients, and for these meaningful subgroups of patients, to develop separate models for each subgroup. So you could see here, for example, clustering of patients according to different ALS progression slopes. And you could see that patient actually, this is just basic clustering without any additional analysis. And you already see that there are tremendous differences between the patients. So what the challenge asks is to take these differences between the patients and identify meaningful clusters, and for each cluster, develop a separate model to predict either how the disease will progress or what would be the survival profile for this patient. One thing else that happened since the last challenge 3 years ago, we also received a lot more data. So this challenge, the first challenge used about 2,000 patients. This challenge would use about 12,000 patients, so much more representative of the diversity of the ALS population. Also, we were very happy to see, so one thing that bothered us is that the data that we have is from clinical trials, and not all ALS patients make it into clinical trials. So what about these patients that never even make it into clinical trials? How could we predict ALS progression for them? So I'm very happy to note that we have a collaboration with 2 ALS registries, one from Ireland and the other from Piemonte in Italy. And they donated data from ALS clinics, about 2,000 patients worth of data that will be integrated into the PROACT database after the challenge. So the challenge would really allow a very comprehensive approach to understanding the diversity of ALS patients. The challenge launches tomorrow. We offer $25,000 in prize and a journal partnership with Nature Biotechnology that have already agreed to publish the results of this challenge. And it allows an opportunity to really bring a first-of-its-kind understanding of how ALS progresses and what can be done to accelerate the research on it. So I invite you all to be part of this challenge and part of its solution. Thank you.
Really exciting. And the next is, if you are looking at data from clinical trials, can you also, on the individual basis, discriminate those who are responders, for example? One of the things that we can also look at, I think, is individual responders to certain therapies. If you have the algorithm for all the rest and you see someone who's not behaving, usually you are using the placebo arm, I think, in most of these databases. But if you're also taking in the drug information, maybe you can extract subgroups of patients who do show something surprising in terms of the algorithms.
So we do have drug information, and also what we see is that the algorithms developed in the previous challenge, so the startup company, Origin, really does a lot of work exactly on that, working with drug companies to understand and maybe their treatment is actually effective for some groups of patients. So can it make it to these patients that could actually use it?
Yeah, because we were discussing individualized medicine before, or personalized, so this is part of it. Great. Any other questions? Yeah.
Do you have any discussion with the regulatory regulatory like FDA or EU, so the endpoints can be, can be accepted for clinical trials, maybe even for the approval on basis on fact basis or something?
Yeah. So we've been working with Origin, who's the startup developed from the first challenge, on really making it reviewed by the FDA. So the FDA doesn't currently force anyone to use any model, but it does recommend certain models. So to really have it reviewed by the FDA and recommended by the FDA. And that would bring it to a lot of clinical trials that might not even be aware of it now or might not use it correctly.
Yeah, but I also
He's talking about endpoints.
Yeah, I'm talking more about the endpoints of the molecular marker. So that it will change, it will like be together with endpoints that are acceptable today, or maybe they will change them.
So the FDA uses the PROACT database. It uses the PROACT database right now not so much to look at endpoints, but to look at how companies plan a clinical trial. So they actually use it in their review to look if their assumptions are correct, if it represents the population correctly. Yeah. Thanks.
Yeah. I just have to cite Russell Katz, who was the previous head of neurology at the FDA. And he said, bring us strong drugs and you will see you don't have to worry about endpoints. So the thing is that when you are getting very subtle, when you cannot really show, but the doctors know how the disease looks and the patients know. And if the drug works very prominently, then it probably shows in the life of the patients. And so thank you, Neta.